Okay, so today's focus will be on genetics, in particular predicting what will occur given certain combinations from your parents. Remember we talked about meiosis and how that allows for the formation of the sperm and the egg cell so they can come together and make new variations. So let's do a little bit of vocabulary review. It's very important. I know in my class we've been really talking about how important vocabulary is in this unit, that we don't mix things up. Okay, so just a reminder as to what a gene is. Okay, so remember, a gene is a section of DNA that codes for one particular protein, right? So a gene is going to be smaller, like this is a chromosome. Okay, so a gene is smaller than a chromosome, smaller than a genome, smaller than the DNA. It's just one little portion of the DNA here that codes for one protein. And that's what we're going to be looking at. We're going to be looking at genes here. And so one of the things we need to remember is that we've got different variations of these genes. And those are called alleles. It's very easy to see in this picture here. Okay, so in this picture here, there is definitely an allele for black hair. Okay, so that's one variation of the gene for hair color, and then there's an allele for brown. So I've got a gene here, so my gene is hair color or fur color on this bear, okay, and my alleles are the different varieties of color that are available. Okay, so that's what my alleles are. They're my different variations on my genes, and they're going to be represented by letters, lowercase and capital letters, big A, little a, big R, little r, and again, we've been talking about that some in my class already. Okay, so for our purposes right now, and next week we'll shift this a little bit, but for our purposes right now, we're going to be focused on what are called dominant alleles versus recessive alleles. Okay, so in a dominant allele, it's represented by a capital letter. Like, for example, down here in this little chart here, subject A has two dominant alleles, both represented by capital G's. And these are always expressed. And by expressed, we mean that you can see them. They're shown. Okay, so these are always expressed. You can always see them. For instance, in this picture with the mice here, okay, the allele, the dominant allele is capital G, and it expresses or shows gray fur. So that means this mouse here has to have some dominant alleles. You have to have some capital G's. The other option here is a recessive allele. And so recessive alleles are represented by lowercase letters, okay, and they are only seen if both alleles are recessive. So for instance, in our diagram down here, where I've got one dominant and one recessive, that mouse still looks gray, okay, because I don't see the trait that the recessive allele codes for unless I don't have any dominant ones at all, and I only have recessive alleles. Okay, and that's where our homozygous heterozygous comes into play. We've talked a little bit about homozygous heterozygous with your vocabulary that we've been working with. Okay, remember, all of our organisms, most organisms that we're going to be talking about are going to be diploid. Remember, they've got two sets of they've got a pair, two sets of every chromosome. Two sets of chromosome number 1, two sets of chromosome number 22. You may hopefully realize this is a female because these two don't match. So there's an X and a Y. I'm sorry, a male. So this is a male karyotype here with the X and the Y chromosome. And so he has one from mom, one chromosome from mom, one chromosome for dad. Those chromosomes are homologous and they code for the same thing. Okay? And so everybody, since they have two copies of all these chromosomes, are going to have two copies of every allele. That's how these mice over here got one from mom, one from dad, one from mom, one from dad. So they get an allele from each parent. And that's where this homozygous heterozygous comes into play. Okay, homozygous, you have the same alleles. Whether they're both dominant or they're both recessive, that doesn't matter, as long as they are the same. Remember, homo is the same. Hetero is different. So with a heterozygous organism, I've got one of each, okay, a big and a little. And those are different. It doesn't matter that they're both A's. Okay, a big one and a little one makes them different. Okay, so heterozygous for a particular genotype would be big A, little a. Homozygous would either be big A, or little a, little a. Okay, so you may have heard me throw these words out a couple times on the last slide. Okay, we do need to sort out in our head what genotype versus phenotype is. Okay, so our genotype is going to be the actual alleles. So when we're talking about genotype, we're talking about the genes. Okay, there's our genotype, my actual alleles. Do I have big G, big G, big G, little G, little G, little G? Okay, it's my actual alleles. I will always have 2. Okay, I will always have 2. Because remember, we're diploid organisms. So I'm always going to have 2 alleles when I'm talking about genotype here. Okay, I could never just say big G is my genotype or little g is my genotype. Okay, I'm always going to have 2 alleles in my genotype. My phenotype is my physical characteristics. Okay, my phenotype is my physical characteristics. What do they look like? That is a T. Okay, what it looks like. So, for instance, here, my phenotype is either gray fur or white fur. Okay, so we need to keep in mind that we always, as a general rule, we're going to have 3 options for genotype. Homozygous dominant. So this mouse here is homozygous dominant, 2 big G's. This one here is heterozygous, a big and a little, and homozygous recessive, 2 littles. Okay, so we usually have 3 options for genotypes, but usually only have 2 for phenotype, because remember, all it's going to take is 1 dominant, okay, to make it look dominant. Okay, and right now we're talking about complete dominance. We're not talking about stripes or polka dots or a lighter shade of gray because it was dark gray and white and now it's light gray because it has both. No. Okay, if there is 1 dominant allele present, then that's what you see. Okay, so as a general rule, we usually only have 3 genotypes, yet only 2 phenotypes for what they could look like. Okay, so now that we've got our vocabulary, handle on our vocabulary, let's take a look at how we're going to do some of these predictions. Hopefully you're familiar with some Punnett squares from middle school, okay, because we're going to go through monohybrid as well as dihybrid squares. Okay, so this here is what we would call a monohybrid square. Mono is 1. Right, so we're only looking at 1 trait. That's the mono part here. We're looking at 1 trait. So we're looking at this gray fur. So I've got 1 mouse that was heterozygous. So let's just call that mom. It doesn't matter who. Okay, let's just say this is mom. Okay, and let's say that this is dad. Okay, so I've got 1 mouse that is heterozygous. Mom is gray. She looks gray, right? Because she has 1 dominant allele. So she looks gray. Dad looks gray, but mom is heterozygous. She carries that allele for having white fur. So if I wanted to predict the possibilities that they could of combinations that they could pass on to their children, I can use this Punnett square. And so that's what the Punnett square is used for. Like I said, to predict possible genotypes as well as phenotypes, because you can, if you know the genotype, you can predict the phenotype. So to set up our Punnett square, first we need to find our gametes. Okay, and so mom has the ability, when mom does meiosis and splits up, she has the ability to pass on either a big G or a little g. So we need to spread those out on the sides. It does not matter who's on the side versus who's on the top. None of that matters as long as you keep all of mom's alleles together and all of dad's alleles together. So this dad has a big G, big G. Those are his only possibilities of things that he can pass on. Big G, mom can pass on a big G, little g. So now all we do in the middle of the square is we determine the combinations that mom and dad could have together. Right? Mom could have a big G, dad could have a big G. Mom can have a big G, dad can have a big G. Mom can have a little g, and we always write the little one second. Dad could have a big G. Okay? Dad could have a big G. Mom could have a little g. And so what we have here in the middle, those are the offspring possibilities. So the middle here, I know it's getting a little messy. We're going to do some practice problems. These are my offspring possibilities. These are the possibilities of my children. On the edges, I have my gametes. On both edges, on the side and on the top, I've got my gametes. And then in the middle, I have my offspring. And if you look over here, The male sperm was carrying a big G. The egg had a little g. So the possible genotype for the fertilized zygote now is big G, little g. So we're just predicting what the offspring could have. So there are 3 ways that we can report our results from our Punnett squares. Okay? We can put them into fractions. Okay? So I can put them into fractions. What I have here is, looking at my offspring, half. Okay? I've got 4 possibilities, right? Okay? 4 possibilities. 1, 2 of them, okay, which reduces to a half, could be heterozygous. Okay, if you have trouble answering probability questions, because most of the time when they're asking you for a fraction, they're going to, a lot of times the wording of the question will want to know the probability that they will have a certain genotype or phenotype. And so when you're asked about probability, y'all know on the bottom, we always put our total. So in this case, my total is 4, right? And on the top is where I answer my question. So in this case, my answer was 2, right? So I had 2 that could be heterozygous out of the 4 total. So that's my fraction of 1/2. I can also represent these with a percentage. Okay, so you could be asked what percent of the offspring will be a certain genotype or phenotype. In this case, I've got 75% of them, these 3 out of 4 right here. So that 75% are going to look like they have gray fur. Because remember, all it took was 1 capital letter to have gray fur. And then the last way is you can use ratios to determine the offspring. Like my ratio here of gray fur to white fur, this one is a little bit different. Okay, but you can see I've got 2 that will have gray fur. I've got 2 that will have white fur. That reduces down to a 1-to-1 ratio. And we're going to look at next how we're going to be able to calculate. Y'all will need to be able to calculate genetic ratio, genotypic ratio, and phenotypic ratio for monohybrid crosses. Okay, so to calculate genotypic and phenotypic ratio, let's look at this example Punnett square. So I've got my gametes of mom and dad, so that means my offspring here are, and I'm going to change this. I'm going to make dad be heterozygous. So my offspring possibilities here are these 4 possibilities for my offspring. And so if I'm asked for my genotypic ratio, okay, my genotypic ratio is going to be the number of homozygous dominant genotypes, because remember it's a genotypic ratio. So my number of homozygous dominant genotypes, and so if I look at this here, okay, that's going to be 1, right? I've got 1 that is big G, big G. So in this case, that would be big G is what I'm looking for. And so my next thing, so I've got homozygous dominant genotypes. So now I'm going to count my number of heterozygous genotypes. Okay, and so I've got 1, 2 for heterozygous genotypes. So I've got 1, 2. So, I was looking for big G, little g. And then hopefully you'll realize your third one is going to be your number of homozygous recessive genotypes. That's my last possible genotype that I can have, right? Little g, little g. So in this case, I've got 1 that can be little g, little g. So my genotypic ratio is 1-to-1 for this particular example. So again, when I'm looking at genotypic ratio, I want the actual alleles, I want the genotypes, and I just want to count how many are homozygous dominant, how many are heterozygous, how many are homozygous recessive. That's all I do. Please don't make it any more complicated than that. Just count how many you see inside of the square that are big G, big G, big G, little g, little g, little g. For phenotypic ratio, now you have to apply these genotypes and think about what they'll look like. Because remember, I'm talking about my phenotype here, my physical characteristics. So with my phenotypic ratio, I'm just going to count the number that look dominant. So in this instance, so in this case here, I've got 1, 2, 3 that are going to look dominant. Okay, so I've got 3